Can take this out. Okay. Right. Good afternoon, everyone. I trust you're ready for your MCQs and you've read done all your bedtime reading from yesterday. Right. So remember, just to recap, we said we would do detection of TB and treatment of TB. And then I'll do a short recap at the end of some of the things that we talked about yesterday. And then hopefully we'll have about 10 minutes to just chat about things in general. Again, just to emphasize that I have put in additional slides. I will not go through every slide and all the material, but I thought these two lectures I gave yesterday and today would make a nice slide pack for you so that you can just kind of flip through. And it's just a good way to refresh oneself of the course material, if you want to. Okay, so we covered these slides yesterday, and this was yesterday's overview, part 1. Well, part 2, actually. This is overview of part 2. So we're going to cover today how to detect TB, what are the drawbacks of current diagnostic tests, and then the treatment of TB, and then a little bit on prevention, mainly talking about vaccines. And then just feel free to interject or stop me as you wish. Now, unfortunately, the detection of TB and diagnosis is a major problem. I told you everything about TB, it's odd. Its history is odd. Its structure is odd. Its division time is odd. And believe it or not, we don't have a simple user-friendly blood or simple urine test for TB like we do, for example, for diabetes. You can do a simple blood test, not only diagnose diabetes but also monitor it. And before we even get to diagnostics, it's very important to understand the public health strategy that we currently use. And that's quite antiquated and illogical. And that's one of the reasons why we have so much of TB. And that public health strategy is one called passive case finding. Passive case finding means that you rely on the patient or the person with TB to actually seek health care, to come to the clinic, to come to the hospital. We call that passive case finding. And it's generally the model we use for many different diseases. Okay. And this paradigm of active versus passive case finding exists for many diseases. Let's take diabetes for the example. About 10% of the population is diabetic, and half of those are undiagnosed, right? So there are many people walking around in the street with diabetes, and what we should be doing is having kiosks all over the place, stopping people and testing them so we can pick up diabetes earlier. If we wait for diabetics to develop symptoms and come to the clinic, it's going to be far too late. The same paradigm with TB. It's not any different for TB than any other disease. The problem with tuberculosis is by the time they come to the clinic and have symptoms that are quite advanced, the horse has already bolted and they have spread about 95% of the disease already. Okay? And so in other words, by the time they get to the clinic and you start treatment, probably 99% of the damage is done already, in the sense that these cavities in the lungs and all these bugs have been coughed up on so many people. And so the question is, how do we take diagnostics out into the community? Most of the TB lies undiagnosed in the community. And there's a startling statistic. About 42% of all TB globally, those 10 million cases I told you about, 42% of them are undetected, right? And these are the cases in the peri-urban slums, in the informal settlements and the shanty towns. And so we did this study. We started this work about 5 years ago. And we published this in a very prestigious journal not so long ago, the Lancet group of journals. And here you can see we have this van, and there's a little tent outside this van, and this is in Langa. at the entrance of an informal settlement, and we loaded Gene Expert, which is a molecular test for TB. It's a DNA detection test. We put it onto this van. We had a generator, and we took this van out into the community. And we showed for the first time, it was a randomized controlled trial. So randomly, when people came, we had loudhailers, we had pamphlets, and when people came to this van, we randomly allocated them to be tested in this van with these new molecular diagnostic tools, or for their samples to be, or for them to just get what was being done all the time, which is smear microscopy, where we looked under the microscope and we waited for the bug to grow. And the bottom line is we found so many more cases using this approach, and we showed for the first time that this was feasible and detected a lot more cases. And the next challenge was, well, this was quite user unfriendly, and so could we actually, let me just see what so could we actually shrink this down? So we managed to get a much more advanced version of this test format. It's called Gene Expert. It's a molecular test format where we detect TB DNA, and it's been miniaturized from quite a sort of a machine, probably as big as this, into quite a little battery-powered unit. And you can see it's all in the back of this Toyota Avanza. There are 2 healthcare workers, and there's the whole setup here you can see on the screen. And so we did a second randomized controlled trial, and we've just finished recruiting. We screened 5,000 people in Guguletu and Langa, and what this showed for the first time is you could scale it down, and this was the first time that we showed you could use modern molecular tools, take it out into the community. So the paradigm now changes, where what our lab is now thinking about is actually moving it even further and self-testing for TB, right? So there's this concept of self-testing for diabetes. You self-test for pregnancy, right, at home. Self-testing for HIV. There's HIV self-testing now. It's just a simple thing. You put saliva on, it detects HIV antibodies, and an app is used and it links you to a counselor. So everything is moving to decentralized diagnosis. So we're moving further away. At the moment, we are very centralized. All the samples from everywhere get sent to a centralized laboratory. It's in Greenpoint or Kruderskir Hospital, right? That's for the whole of most of the Western Cape. And so one paradigm is to take these diagnostics, stick them onto mobile clinics, take them out into the community. But can we move this paradigm to creating a test format that is available at health kiosks, at shops, supermarkets, pharmacies, anywhere, Checkers, Pick and Pay, out in the communities. And so this represents a new paradigm for TB testing. And in fact, we are doing some research here at UCT and through our lab, and we've discovered using mass spectroscopy a group of TB-specific proteins in the urine. It's a patent currently owned by UCT, and I'm a co-inventor on that patent. And we are using this now to develop a very simple urine-based test, very much like a urine-based pregnancy test, where you can make a diagnosis of TB. We've just got a very large grant from the Department of Trade and Industry to develop such a platform. So that's kind of where we should be heading to. But that's where we're heading to. What do we have right now? Well, right now, I've just kind of configured this slide. This is just an overview of the different ways to diagnose TB. You can look for the bug under the microscope. The problem is you only see it in about 50% of cases, and that result is available the same day. Alternatively, you can grow the bug, but that takes 4 to 6 weeks, takes very long. You can detect antigens from the bug in urine, for example. There is one urine-based test now, but it detects a molecule called LAM, which is part of the cell wall of TB. but it only works in advanced HIV. It doesn't really work in everybody. So, and that's why we are working on this new test, which looks at lots of different biomarkers. And number 4 is the main way we detect TB right now, which is to detect the DNA of the bug in the sputum. So it's a DNA amplification test, and a sputum sample is donated at the clinic, and that goes to Greenpoint or to Kruderskir, and this is stuck into a machine which extracts the DNA of the bug. It's a genetic material of the bug. It amplifies it, so it's very sensitive, and it detects it with a probe. And that is number 4 is, so if you walk into any clinic or any hospital in South Africa, whether it's the public or the private sector, this is the frontline test we are using right now. It's called the Gene Expert. And the other TB tests is number 5, where you can detect an immune response to TB. That's more for the diagnosis of sleeping TB, and I'll talk about that just now. Number 6 is to detect antibodies to TB. There were sort of these test formats that were being punted around 15 and 20 years ago. None of them work, and so we don't use these kind of tests anymore. And then lastly, there's been work on detection of volatile organic compounds. It's sort of like an electronic nose. There's a lot of work done on it. We did a big study recently for a company in Europe, but it doesn't really work as well yet. The technology is not quite there. So the DNA detection tests is what we have started using now. We've only started using it in the last 5 years, and before then, all the diagnostics we had were over 100 years old, like this car you see, and nothing has changed for over 100 years till about 5 or 6 years ago. This is just, again, pictures of what it looks like under a microscope, and when we grow the bug, what it looks like. And when we grow the bug, we can expose the bug to different antibiotics, and we call that drug susceptibility testing. So we can see whether the bug is sensitive to a particular antibiotic. And we do that for lots of different infections. It's not unique to TB. So here's a picture of this Gene Expert platform I was talking about. It looks like this. You get a sputum sample, you add some buffer, you stick it in a cartridge, you put this cartridge into the machine, and it's an automated format. It extracts the DNA, it amplifies the DNA, and it detects TB. It tells you TB or no TB, and it also tells you if it's drug-resistant TB, because it will pick up a mutation that affects the key TB antibiotic rifampicin, and so it tells you whether it's drug-resistant or not. So that's the big advance over the smear is that previously we only could detect 50% of TB the same day. Now we can detect about 85% of TB the same day. That still means about 15% of TB still requires culture, and the diagnosis takes several weeks, and you think, well, that's quite straightforward. It's not, because you can only culture something if you have a sample. And the big problem in TB is not everybody coughs up the spit, right? The TB is in the lungs, and what are we sampling? We're sampling the spit, okay? So the big challenge in TB diagnosis, we're working on it, and there are about many other labs throughout the world working on this. The big challenge is to develop a non-sputum-based test for TB, using, for example, exhaled breath or urine, where you can always access a sample even in children, right? Big problem in children, how do you make a diagnosis? You can't get any sputum. You can't ask a child to cough up sputum, right? Very difficult. So that's a major challenge to TB diagnosis at the moment. Now, remember I was talking about latent TB infection yesterday? Sleeping TB, and I told you we don't even know what it looks like under a microscope. We can't see it. And we actually don't understand why we can't see it. It's very strange. Nobody knows this. And people have suggested that there are cell wall free forms. So this bug, when it goes into a hibernating state, maybe it loses its normal cell envelope that we stain. And it's also immunologically invisible as such, because if we could see latent infection, we'd see a lot of inflammation, right? We can look under the microscope, We can look at cells, we can see cancer cells, we've got very good microscopy tools. But when you look at these tissues where they're supposed to be sleeping TB, you don't see them, you don't see anything. So they are invisible, and that remains a mystery. One of the mysteries of science. Now, that begs the question then, how do we diagnose something you cannot see? Okay, and one way is to use a tuberculin skin test, and tuberculin is simply a mishmash of TB proteins, and we inject it into the skin. Some of you may have had this test or seen this test. It's called the tuberculin skin test, and if they put it into your skin, and if there's swelling at the injection site where you inject this, it means that your body's immune cells have seen TB or TB-like antigen in the past, and that's why you get the swelling. That's all it means. And that's a big catch, TB-like, because I told you yesterday, how many different species of mycobacteria are there? At least 150 that we know of. There are many others, right? So it starts getting quite fuzzy and inaccurate because there's a big overlap in antigens between TB and all its cousins, including bird TB and various other forms of TB. So this test is quite sensitive because it tells you that, hey, this person has seen TB before, but many of us have seen TB. Doesn't mean we have sleeping TB. So an advance on this has been some blood tests. One of them is called quantiferon, right? Some of you may have heard of TB blood tests. Anybody here heard of TB blood tests? Pick up your hands. No one heard of TB blood tests. Okay, that's very good. Because if you hear about it, you can tell someone, well, Prof. Data told us we cannot use this to diagnose active TB. Right? It's a test for sleeping TB, and we don't really use this test here because we don't really look for sleeping TB in South Africa. And the reason for that is we got enough trouble with active TB, 500,000 cases a year, to worry about sleeping TB. And even if we treated and found sleeping TB, people would get infected again. So we've just forgotten about sleeping TB. But I'm just telling you that there are these blood tests. Now, there's a more advanced skin test where they've actually isolated very specific TB antigens, and we've done a lot of the clinical trials here. It's currently undergoing registration with the EMA, but it's just a sort of a luxury version of this test, but all basically have the same drawbacks. You cannot really prove sleeping TB, just infer that someone's more likely to have sleeping TB. I've already spoken about the ideal diagnostic test, and we did a big study a couple of years ago where we took this Gene Expert machine, and instead of leaving it in Greenpoint, remember I was telling you about getting diagnosis closer and closer to the patient. Where are the clinics? The clinics, Langa Clinic, Guguletu, Weinberg Clinic, are all very near people. So we took this diagnostic out of Greenpoint, we stuck it in the clinic, and we said, is it feasible just for a nurse, right? We gave a very basic training, and we found that this machine was very advanced, very automated, as I showed you, and it was quite feasible for a nurse. The results the nurse got were exactly the same as the lab technicians got. So we are not really exploiting the portability of this technology, right? At the moment, we can have it in all our clinics, but we don't do so. And why is that important? Because it just turns out about 25% of people who come to test at the clinic, they send their spit off, it comes back positive. They never come back to get their result. And if you try to call them, you can't get hold of them. They give different numbers, different addresses, and we'll talk about that later on. Okay, so as I said, it's all good and well. We're making the diagnosis more quickly with these new tools. But really, if you stand back and you look at it, it's not a big advance because all the damage is done already, right? And that's what we're showing in this trial. You can see you got the pink bars and the blue bars. The one is smear microscopy, the old test, and the blue bars is Gene Expert. And you can see that this is days into, after they arrived at the clinic. And you can see even at 5 days, there's not a big difference between the two bars. And the reason is that many of the doctors will take an educated guess looking at the chest X-ray and examining the patient. Bottom line is, by the time you get to the clinic, all the damage is done. And so while it seems that, we have this fancy DNA tests, right? that can now pick up the TB DNA, it's really a toothless bulldog, because it's not really getting at the heart of the issue, which is making the diagnosis very early when TB develops out there into the community. So let's go on to treatment of TB. Okay, so we've finished on the diagnosis. We're going to the treatment of TB now. And I already told you a little bit about this yesterday. There was, in the 40s, there was a guy called Waxman who discovered streptomycin. Streptomycin is an antibiotic made by a fungus. Many antibiotics are made by fungi. And this guy was called Lehman, who discovered another TB drug called Paz, which we now using only for drug-resistant TB. And they found that when they gave streptomycin, they thought, geez, now we've solved the TB problem, but it was only moderately effective. And when they used Paz as well, they used these drugs alone. They found it was only moderately effective. And here you can see that this is the percent of bugs that grew. And you can see when you look at this after the months of treatment, you only got an effect if you combine both the drugs. Okay, so this was the first lesson for people looking at TB treatment, that you had to combine two drugs to get an effect. If you just used one antibiotic, it didn't really work. So why do we need combination therapy, right? You treat pneumonia, you generally need one antibiotic. You have a urine infection, you need one antibiotic for 3 to 5 days. Why is it with TB that we need so many drugs? Okay, and the reason for that is to prevent drug resistance. Okay, so it just turns out, and this is what this figure is trying to show here, is by chance there are TB bugs that are resistant to a specific antibiotic by chance. So when they use streptomycin alone, it was effective for a short while, and then what it really did, it killed all the bugs that were sensitive to streptomycin, and the few that remained in the body that were naturally resistant to streptomycin then started growing up. And it just turns out with TB, their structure and their genetic makeup is such that 1 in 10 million or 100 million are naturally resistant to a specific antibiotic. And so you have to combine antibiotics when you treat TB. I told you also it's a slow-growing bug. So unlike bugs that, for example, cause food poisoning, they divide very rapidly. The antibiotic kills them very quickly, and so you can get a rapid death of these bugs. And if you have killed all your bugs quickly, you have no bugs, you can't develop resistance, right? So all your bugs are dead. This bug is a slow divider, takes longer to kill, and that's part of the reason why you need combination therapy. Now, the other problem with TB is you need 6 months of antibiotics. That's also quite curious, hey? Why do you need 6 months of antibiotics? And the reason for that, in fact, is that you have different populations of bugs, TB bugs. Now, I told you everything about TB is odd and peculiar. And one of the peculiar things about TB is that there's different subpopulations of bugs. There's a big population that grows rapidly and divides rapidly, and that's killed rapidly by antibiotics. There's another subpopulation that tends to persist in acidic conditions, and so you need a special drug for that little population, because if you kill out most of the bugs, it's those that remain will grow up again. And it just turns out with TB, and again, we know very little about this, there are these what we call not resistant but hardy population of TB bugs. And these guys are called long-term persisters. They can withstand, they have some mechanism where they can withstand antibiotics for many, many weeks, but not many, many months. And we call them, there's a small population, they call them long-term persisters. And they're a very strange group of bugs, TB bugs, and they have slightly different staining characteristics if you use special stains. And again, it's one of the enigmas of TB, right? This mysterious population of long-term persisters, not very well characterized. We don't know how to identify them, but we know they exist. And that's the one big reason why you need therapy for 6 months. You can treat for 2 months, and you probably will cure probably 60 or 70% of TB patients, but about 30%, 1/3 of patients will relapse when the TB comes back. And when it comes back, it can affect the brain, they can die, damage the lungs more, disastrous consequences. We don't want that. We're happy to tolerate maybe maximum 5% relapse, okay? 30% is too high, so that's why you need long therapy. Others don't agree with that view. They say, well, these long-term persisters, we can't really characterize them. We don't know what they look like. We can't identify them. Do they really exist? Is it really a figment of our imagination? And the real reason why we need prolonged therapy is because of immune paresis. Now, when you treat an infection like a urine infection or a pneumonia, you just don't need the drugs alone. You need the immune system, right? It's not just the antibiotic. The antibiotics help to kill the bugs, but you also have an immune system, and the same with cancer. So a lot of people think that TB corrupts the immune system. And we're not going to talk about that in these lectures because that's people have entire conferences for 5 days talking about these things and how the TB bug cripples and paralyzes the immune system. But it can turn on, for example, groups of cells called suppressor cells, okay? And think about it, if you were a TB bug, you'd want to cause maximum inflammation and damage to the body and actually drive the formation of these cavities in the lungs so you can spread, right? And so really, TB plays havoc with the whole body's immune system. It accelerates an inflammatory response that doesn't kill the bug but just damages tissues. It hides away, it turns on suppressor cells and all sorts of funny business. And others say that, look, that's the real reason why we need, because the immune system takes so long to recover. So that's the reason why, or some of the reasons why we need antibiotics for several months. And then how do we prevent resistance? That's another thing, is that I told you that by chance there are so many, there's groups of these bugs that are naturally resistant to antibiotics. So when we use combination therapy, then that helps to prevent the amplification of resistance. But it's not that simple, because I told you about these TB cavities, and it just turns out that drugs don't penetrate these cavities very well. Remember yesterday I told you that this is a niche for the TB bug. They hide away in these cavities in the lung because the drugs, remember these cavities are just holes in the lungs. They're avascular. You got this soup of very nutritious stuff sitting in this cavity, and they're all growing there, and the drugs can't get in there. The reason the drugs can't get in there is you need a vascular supply to deliver the drugs, okay? And it's got a thick fibrous wall so the drugs can't penetrate. And in fact, we published a paper recently showing for the first time that what this graph shows is that as you get to the center of the cavity, this is the outside of the cavity, this is blood and the outside of the cavity, and you can see this drop, this sudden drop here. You can see the drug level on this side, and just to simplify this little inset for you, here you can see good levels of drug in blood, in the normal lung tissue, in the cavity wall, and as you get to the center of the cavity, there's hardly any antibiotic, okay? And that's one of the big problems with TB. And the way we showed this was on people we can't cure with TB who have drug-resistant TB, and we can't cure them. Our last result is chop out the lung. And we do that every month or two at Critics Care, and we take those lungs to our lab. So for the first time, we could actually get the lung, we could measure the drug levels in the cavity. And we found for the first time that at each of these sites, when you measure actual drug levels, it's extremely low in the center of the cavity. And this is one of the ways in which we get what we call resistance amplification, or the development of drug-resistant TB. Up to now, we thought, we blamed the patient. We said, these guys aren't taking their drugs, the doctors and the nurses aren't doing their job, they give the wrong drugs, the health system doesn't work properly, they can't get the drugs to the clinics, and all this type of things. And we kept blaming the system and the patient. But we now realize that's only part of the problem. A big issue is actually getting the drugs into these cavities, and that's one of the big reasons why we develop resistance. And why is that? Well, if you have very low drug levels, if you get any bug being exposed to low levels of drug, they develop resistance. How does that happen? Well, it's evolution. They turn on genes that actually can pump drugs back out of the cell and all sorts of things, right? Again, that's a whole nother talk, just to simplify it for you. And this I just put into your slide pack. This slide shows you the mechanism of action of different drugs. So for example, rifampicin is the main TB antibiotic, and what that does is it stuffs up an enzyme that generates RNA. RNA is the template from which we make proteins. And so basically that just gives you one example, but all these drugs will really hit at a vital component of the TB cell, whether it's to make RNA or to synthesize protein or the cell wall or whatever. We're not going to go through the mechanism of each drug. Just the main message is that TB drugs, anti-TB antibiotics, work by really sabotaging key components of the TB bug. Now, another big problem we have is, if you thought 6 months was long, if we treat drug-resistant TB or MDR TB, which is resistant to the two main TB antibiotics, we need to treat for 18 months. That's a long time. Okay, and up to now, you got painful injections for 6 months and no local anesthetic. And is it surprising that half the patients don't come back? Right? It's a terrible treatment to take. Even the course for 6 months has a lot of side effects. People feel quite nauseous, makes you feel unwell and ill. It doesn't make you feel nice. And here's this is all the medicines for drug-resistant TB, the different antibiotics, okay? And then you got somebody who also has HIV. Remember, I told you 50 to 60% of people who have TB are HIV infected. And look at how many pills they have to take every day. It's really surprising that they don't want to come back for treatment, that they cut their treatment short. And so here you can see, if you had to stack up all these pills, in a year, you'd actually equivalent to the height of the Golden Gate Bridge in San Francisco. And this is the actual case that we had not so long ago. The 46-year-old person with TB turned out to have drug-resistant TB, and he was actually a clerk with a higher diploma, so this is not an uneducated person. There's her chest x-ray here. You can see the lungs so those are the breast shadows. That's the heart in the middle, and all these white bits here that you can see, it's not normal. It should all be black. And those are all really pneumonia, and you can see a little hole in the lung here. And his wife was concerned because he wasn't really getting better, and then she found all these tablets in his drawer and been keeping all of them, and he felt awful. He didn't want to take the drugs, and of course it didn't help when the Sangoma told him that he should not worry because he will be cured. Okay, so it's a very multifactorial, difficult problem. This is more the human side of managing people with antibiotics and drugs. In the real world, this is what happens. And this was in a research study that we were doing. And you can imagine what happens. Here we had dedicated people to support the patient, check on them and all that. It's actually much worse in the real world. And I already mentioned the adverse events of the drugs. And so there's so many challenges in TB. There's this problem of delayed diagnosis that I told you about. Firstly, before you even go there to even get a sample, because people can't cough up the sputum, that's a problem. You have patients with complex needs. People need to go to school. They need to pay their mortgages. They need to work. They can't come to the clinic every day. You need to take treatment for several months, maybe 18 months if you've got drug-resistant TB. A lot of people cough. They cough a little bit. They don't even know they have TB, and they go on like this for months. And there's a lot of stigma attached to TB. People don't want to be diagnosed with TB. Sometimes that's why they don't never come to collect their results, okay? And they go to different clinics expecting a different diagnosis. There's also a very under-resourced service now with the advent of HIV and this massive burden of TB. So there's so many different angles to this whole thing. This is something called video DOT. So at the moment we use directly observed therapy, where we assign either the clinic or somebody, a shopkeeper or somebody, a teacher, who can actually on a daily basis dispense the drug and actually see the patients taking their drug, because adherence is a big problem. And this is just something you see in Europe and the US, paradoxically, where TB is the least of their problems. With technology and smartphones, you're looking at a video dot directly observed therapy. The problem with using the system here is that, with everybody's got a cell phone here, but data is so expensive in South Africa, right? And bandwidth is a problem. And so that's another example of how mismanagement and corruption has impacted us. I mean, why is cell phone data so expensive here? There's no good reason for that, right? it's a fraction. I mean, I just gave a lecture in India last year, and it's you can phone anywhere in the country, and I think it's one rupee a minute anywhere, and people have data they can transact. It's very expensive. Data is very expensive here, so it's difficult for us to use these kind of methods here. I already talked about the problems with drug-resistant TB therapy. It's a long duration. You need to give people injections and all types of things. And the reality is, it's quite startling. Only about 20% of the world's population can access MDR treatment. You go to countries north of our borders, can't get MDR treatment. The drugs are very expensive. In fact, treating drug-resistant TB is very expensive. At the moment, it's so expensive that although MDR TB and XDR TB makes up less than 5% of our burden of disease, it's already eating up 65% of the total TB drug budget and about half the total TB health budget. It's very expensive. And the reality is that only about a third of people in the world can even access the special diagnostics required to make the diagnosis of MDR TB. So that's the reality of the world. We have 2 new TB antibiotics after a dearth of anything new for almost 40, 50 years. Those are called bedaquiline and delaminid. You may have heard about them in the newspaper. So we are using this new antibiotic called bedaquiline, and that significantly improved treatment outcomes. And so for the first time now, here you can see the success rate is the green one. You can see in South Africa, our treatment success rate for MDR TB is only 50%. And now we have this new drug, bedaquiline, and we're using it. So we at least now, from this year, we're not using injections anymore. And we've shortened the treatment duration to about 12 months. But this is a clinical trial we're doing, and this is all funded from South Africa. And we're the only one in the world doing this, is try and shorten the treatment to 6 months. And I told you need multiple drugs, and because these newer drugs like bedaquiline are so much stronger and more potent, the question is, can we use all these stronger drugs and shorten the treatment from 18 months? South Africa's taken a bold step, first country in the world to go to 12 months, but we're saying, well, let's try and see if we can do it in 6 months. So we have this big clinical trial. We finished recruiting almost 110 patients for this trial. We're in the follow-up phase now, so hopefully in a few months' time we'll be able to have a positive result and tell the world that, hey, you can actually shrink the treatment down to 6 months, no injections, and at least it'll be a major advance. It'll be the same duration of treatment as drug-sensitive TB. Of course, there are trials going on drug-sensitive TB as well, where they're trying to shorten the treatment duration from 6 months to 4 months. About 2 or 3 years ago, there were 3 large studies that were done in the world. They spent almost $1 billion on all the 3 studies, massive studies done in the world, and they failed to shorten treatment from 6 months to 4 months using moxifloxacin, a fluoroquinolone antibiotic. So that was a big failure, and one of the reasons it failed, I showed you the moxifloxacin levels in the cavities are very low, right? But hopefully we have more luck with bedaquiline and delaminid, and there is a big study being done by the Gates Foundation now, where they're using these much more potent drugs to try and shrink the treatment from 6 months. I think they're trying to do it to 2 months now. So I think as technology moves on, we're going to get better and better drugs and be able to shrink the treatment duration. However, as I told you, by the time we treat the patient or even wait for the patient to make the diagnosis, it's too late, okay? So what we really need to do is to prevent TB. Prevention is better than cure. And I showed this slide yesterday already, that if we really want to impact TB, it's not about finding new drugs. It's not about necessarily, taking the diagnostics out into the community will help, but we need to address things like poverty, overcrowding, HIV, indoor air pollution, outdoor air pollution, housing and ventilation, smoking, better nutrition, right? These are the things that are really going to make a big dent in tackling TB. People need to be much more aware of the symptoms of TB. We need to take these diagnostics out into the community. The way the system we're using right now, many of you will remember the old X-ray vans, that went out into the community. They just X-rayed everybody. We need to get that back. There are artificial intelligence algorithms now where you can, where you can have these vans that go out. People will still have to walk up and get the X-ray done. But then what they'll do is they'll just upload this into a cloud, and the computer algorithm will look at the whole thing and say, oh, this one, it's almost like a heat map of a probability heat map of TB. And so that's the way we can use, artificial intelligence to help us. It'll only help. It's not can't do everything we needed to do. It's like the veld fires and the mountains and whatever. You got satellite images and all that, and you need to trial these things and tell intelligent systems exactly what to look out for and so forth. So this is the way in which we need to tackle TB. Obviously, we need to, by finding people early in the community, then we can treat them and then rapidly abort transmission. Now, what about other aspects of prevention? I told you about, a very common sense fact is that people transmit TB through the air, right? They cough, and there's cough aerosol. So if we can interrupt the spread, we should be able to really deal a death blow to TB. But where is this transmission occurring? Where is the spread occurring? Is it occurring in the household? Where is it occurring? And by analyzing the genetic code of TB, we can actually measure very nicely where the spread is happening. And so there's a whole slew of studies over the last decade that have showed that most of the transmission is not occurring in the house, it's occurring outside the house. Where is it occurring? It's occurring in congregate settings, occurring in buses, taxis, shebeens, churches, schools, right? So ventilation, proper ventilation, cough etiquette, awareness of symptoms is quite critical, right? And people are looking at all sorts of innovative little things. You can imagine in Cape Town, especially in the winter months, with taxis, all the windows are closed, people's homes, schools, the workplace, right? So we have a number of cases all the time of screening people for TB who've been exposed to index cases of TB. Another way to prevent active TB is to treat sleeping TB, because I told you that about 5% of sleeping TB will reactivate later on, right? So this is another thing about the TB bug that's quite intriguing. Even if you manage to do all these things and get rapid diagnosis, and you have all these little urine tests out in the community and you can make a quick diagnosis, there's the sleeping TB, and that's like a ticking time bomb, right? That's like almost like the bug's insurance policy where it's going to reactivate later on. So if you actually model this, and if you model this using a computer program, what the model will tell you is no matter what you do, if you don't tackle these sleeping TB cases, you're always going to have this pool, this reservoir of cases popping up all over the place, and then you go through the whole paradigm again. So if we really want to eradicate TB globally, we're going to have to treat sleeping TB, and we don't treat sleeping TB in this country. One of the reasons we don't treat sleeping TB so much is that you need 6 to 9 months of an antibiotic to treat sleeping TB. And we're getting shorter and shorter regimens. There's some antibiotics that have a long half-life. And we think now that we can get it down to about 3 months. And very recently there was a study that got down a regimen to about 4 weeks to treat sleeping TB. It's still a long time, to take antibiotics. Again, even if you treat for a period of 4 weeks, the big problem we have in South Africa, there's so much of TB floating around. You eradicate the sleeping TB, you get infected again. And HIV-infected people are exquisitely sensitive to getting sleeping TB. And so at the moment, that's the only group, and children under 5, that we target for sleeping TB. We don't treat anybody else for sleeping TB. And so people are thinking now of cycling antibiotics, maybe 4 weeks every year or something like that. So that, and this is in high-risk people like HIV-infected people and children. So these are all the challenges that we have with TB. But really the one thing that we really need if we want to strike a death blow without, it's going to take decades before we eradicate poverty and overcrowding. And at the rate we're going, we're actually moving backwards, right? So TB is not going to go away in Africa or South Africa anytime soon. And what we really need from science is a vaccine, an effective vaccine for TB. And the current vaccine is BCG. I told you that a relative of the main TB strain is Mycobacterium bovis. It's just a few genes that have been deleted, and it causes disease in animals and causes a disease in humans exactly similar to the Mycobacterium tuberculosis strain. And then they took this M. bovis strain and they passaged it from culture to culture to culture. And as you passage things, they can lose some genetic elements. And eventually they passage down to the strain, which has got a couple of genes that were lost from a virulent strain, and it's called BCG. So it's got about a 99.9% homology with Mycobacterium tuberculosis, and this is the one that we've all been vaccinated with. Now, this is also quite intriguing. Why doesn't the vaccine work? Now, it's very strange because, in fact, it does work in some parts of the world, but it doesn't work in other parts of the world. It's very, very strange. And studies, and over the last several decades, there have been many big studies, right? I mean, TB was still a big killer in Europe and America in the 40s and the 50s, and a lot of big trials have been done. It just turns out that BCG was very effective in countries. Which countries? UK, very effective. Scandinavia, very effective. Alaska, very effective. And it just turns out that if you looked at trials in India and Africa, the closer you came to the equator, the less effective the vaccine was. Very intriguing. And we don't really understand why. Why is it that, and that doesn't happen with measles vaccine, why on earth does it happen with BCG? And there are a couple of theories on why this happens. The big theory is that, remember I told you TB is one of 150 or 200 species of related bugs called mycobacteria. And a lot of these mycobacteria live in the environment. They're found in soil, they're found in water. You open your tap, and your geyser's probably full of mycobacteria as well. And so these things are everywhere. So when one is born, you immediately exposed to these mycobacteria. They're everywhere. We've evolved with them, okay? And so one of the first things our immune system sees when we're born is mycobacteria. And one theory is that by the time we give BCG, the immune system's already familiar with these mycobacteria, been evolving. They recognize them as old friends. And all these common bugs that we see when we're born, we actually needed to educate our immune systems. And when the immune system sees these bugs, this is one of our old friends, mycobacteria, don't need to worry about this fellow. And so when we give the BCG, the one theory is that, in fact, the response is masked. And in fact, because we've already developed antibodies to all these other mycobacteria, when BCG is actually given, it prevents the replication of BCG. BCG is a live vaccine. When you give it, you want it to replicate in the body and the body to have a good immune response to it. So that's the theory about why we think BCG doesn't work. In fact, the effect wanes by the age of 10 or 12. It does protect against some severe childhood forms of TB. But in South Africa, it doesn't work after age of 10 or 12. It doesn't really work. An alternative theory is that BCG, remember I told you it's lost certain critical genes, and maybe these are the very important genes that produce these virulence factors. And so it doesn't really have the elements in it that's supposed to induce the correct protection. Be that as it may, we're really not sure why BCG doesn't work. Another whole argument to BCG is how you give it. If you give it in the skin, you have all these factors playing out. It just turns out there have been lots of studies done in macaques, in monkeys and rabbits and guinea pigs, and it just turns out if you give BCG via the lung route, if you inhale the vaccine, you get much better protective responses. And after all, you inhale TB. Now, in humans, whether inhaled BCG will be effective, we don't know. We've never tested it. And we've just submitted an application yesterday to the Wellcome Trust to actually test this. We want to test it. We actually want to take BCG. We've over the last 4 years optimized the whole bug load and everything. We take a bronchoscope, we stick the BCG into the lung, but we're going to give it inhaled. We've worked out a way to do that. And so we're going to test it. We want to test it. Well, that's if we get the money. We will ask for £4 million to do the study, to actually test. Right, let's just do this thing once and for all. Let's stick it into the lungs. Does it really protect you? So we don't even know if we're giving it via the correct route. Okay, so these are all the unanswered questions. Having said that, there are a number of new vaccine candidates. This is just a slide showing you preclinical studies. Then they have to establish safety in man, that's phase 1. Then early trials where they test it in small groups of people. But there are two big problems now. You've got all these vaccine candidates, about 20 of them, right? Don't worry about this one. It doesn't really work. So we've got all these fellows here. And just a few weeks ago, there was a big study done on one of these vaccine candidates which showed about 50% protection against active TB, which is not great. Having said that, it's an early phase study. But now there are two big problems with these studies. The first problem is that to evaluate each of these candidates, to develop it and take it through all the clinical trials, roughly, cost is about $1 billion per candidate. Who's going to pay for that? Very expensive. And TB is a poor man's disease. Will a company ever recoup their investment, for a vaccine that's given freely to the whole world? No company does that science. They don't invest in doing that R&D. The second major problem is, even if we have to say, okay, for the public good, what's $1 billion? Not a lot of money in the world today, $1 billion. And could easily be put up by the European Union and the US, and maybe some people say, well, they should do it. It's a trillion-fold higher that is actually taken out of countries like Africa and Asia, so that will be a good reinvestment. But even if we had that money, how do we test these things in small numbers of people? The big problem with TB vaccines, you stick them into a person, right? You need to test thousands and thousands of people, and then you have to wait many years to see if they get TB. So if you're testing small groups of people, you need some signal, some clue that the vaccine is doing the right thing. What is that clue? It's usually a test, some kind of immune test or a blood test or some test, right? For example, if you had to test a pneumococcal vaccine for pneumonia or a measles vaccine, if we make a new measles vaccine, what do we measure? We measure measles antibody levels, right? We can measure it in the blood and say, this vaccine looks very promising, very high titers of antibodies to measles antigen number 64. We don't have such a thing for TB. We don't know what's a good TB vaccine. We don't have a biomarker or a blood test or a specific antibody that we can measure to say, hey, this particular vaccine looks really good. This is the one we should test. We don't have that. We don't know. And these